Hi, welcome to my channel and welcome to the part 2 of the topic photosynthesis. I'm so glad that you're here. So we will continue discussing photosynthesis. So in the previous video, I have introduced you to photosynthesis and I have discussed the processes there, and in this video we will continue discussing how the two different ways of electron flows. So let's go.
During the light reactions, there are two possible routes for electron flow, the cyclic and linear. Linear electron flow, also known as the primary pathway, involves both photosystems and produces ATP and NADPH using light energy.
A photon hits a pigment and its energy is passed among pigment molecules until it excites P680. An excited electron from P680 is transferred to the primary electron acceptor. We now call it P680+. P680+ is a very strong oxidizing agent. So, water, H2O, is split by enzymes and the electrons are transferred from the hydrogen atoms to the P680+, thus reducing it to P680. Oxygen is released as a byproduct of this reaction. Each electron falls down an electron transport chain from the primary electron acceptor of PS2 to PS1. Energy released by the fall drives the creation of a proton gradient across the thylakoid membrane. Diffusion of H+ protons across the membrane drives ATP synthesis. In PS1, like PS2, transferred light energy excites P700, which loses an electron to an electron acceptor. P700+, or P700 that is missing an electron, accepts an electron passed down from PS2 via the electron transport chain. Each electron falls down an electron transport chain from the primary electron acceptor of PS1 to the protein ferrodexine. The electrons are then transferred to NADP+ and reduce it to NADPH. The electrons of NADPH are available for the reactions of the Calvin cycle. This process also removes an H+ from the stroma.
This is a mechanical analogy for linear electron flow during the light reactions.
Cyclic electron flow uses only photosystem 1 and produces ATP but not NADPH. There is no oxygen that is released. Cyclic electron flow generates surplus ATP, satisfying higher demands in the Calvin cycle.
Cyclic electron flow is thought to be evolved before linear electron flow. This flow may protect cells from light-induced damage. So some organisms, such as purple sulfur bacteria, have PS1 but not PS2.
Chloroplasts and mitochondria generate ATP by chemiosmosis, but use different sources of energy. Mitochondria transfer chemical energy from food to ATP, while chloroplasts transform light energy into the chemical energy of ATP. Spatial organization of chemiosmosis differs between chloroplasts and mitochondria, but also shows similarities. In mitochondria, protons are pumped to the intermembrane space and drive ATP synthesis as they diffuse back into the mitochondrial matrix. In chloroplasts, protons are pumped into the thylakoid space and drive ATP synthesis as they diffuse back into the stroma.
ATP and NADPH are produced on the side facing the stroma, where the Calvin cycle takes place. So in summary, light reactions generate ATP and increase the potential energy of electrons by moving them from H2O to NADPH. The Calvin cycle, like the citric acid cycle, regenerates its starting material after molecules entered and leave the cycle. The cycle builds sugar from smaller molecules by using ATP and the reducing power of electrons carried by NADPH.
The Calvin cycle has three phases. Phase 1 is the carbon fixation catalyzed by Rubisco. Phase 2 is reduction. Phase 3 is the regeneration of the CO2 acceptor.
Carbon enters the cycle as CO2 and leaves as a sugar named glyceraldehyde 3 phosphate. For net synthesis of 1G3P, the cycle must take place 3 times, fixing 3 molecules of CO2.
Dehydration is a problem for plants, sometimes requiring trade-offs with other metabolic processes, especially the photosynthesis. On hot, dry days, plants close their stomata, which conserves H2O, but also limits photosynthesis. The closing of the stomata reduces access to carbon dioxide and causes oxygen to build up. These conditions favor an apparently wasteful process called photorespiration.
In most plants, C3 plants, initial fixation of CO2 via Rubisco forms a 3-carbon compound called 3-phosphoglycerate.
In photorespiration, Rubisco adds oxygen instead of carbon dioxide in the Calvin cycle, producing a 2-carbon compound. Photorespiration consumes oxygen and organic fuel and releases carbon dioxide without producing ATP or sugar.
Photorespiration may be an evolutionary relic because Rubisco first evolved at a time when the atmosphere had far less oxygen and more carbon dioxide. It limits also the damaging products of light reactions that build up in the absence of the Calvin cycle. In many plants, photorespiration is a problem because on a hot, dry day, it can drain as much as 50% of the carbon fixed by the Calvin cycle. C4 plants minimize the cost of photorespiration by incorporating carbon dioxide into 4-carbon compounds in mesophyll cells. This step requires the enzyme PEP carboxylase. PEP carboxylase has a higher affinity for carbon dioxide than Rubisco does. It can fix carbon dioxide even when carbon dioxide's concentrations are low. These 4-carbon compounds are exported to bundle sheet cells where they release carbon dioxide that is then used in the Calvin cycle.
In the last 150 years since the Industrial Revolution, carbon dioxide levels have risen greatly. So the increasing levels of carbon dioxide may affect the C3 and C4 plants differently, perhaps changing the relative abundance of these species. So the effects of such changes are unpredictable and a cause for concern.
Some plants, including succulents, use crassulacean acid metabolism to fix carbon. CAM plants open their stomata at night incorporating carbon dioxide into organic acids. Stomata close during the day, and carbon dioxide is released from organic acids and used in the Calvin cycle. As for example, in these two pictures here, a sugarcane and pineapple, in the sugarcane, it involves a spatial separation of steps, while in the pineapple, it involves temporal separation of steps.
The energy entering chloroplasts as sunlight gets stored as chemical energy in organic compounds. Sugar made in chloroplasts supplies chemical energy and carbon skeleton to synthesize the organic molecules of cells. Plants store excess sugar as starch in structures such as roots, tubers, seeds, and fruits. In addition to food production, the most important thing here is photosynthesis produces oxygen in our atmosphere, in which we humans need to stay alive. So we need plants like trees or make our environment green to make the air we breathe clean. So that is all for this video. It is all about photosynthesis and how it is important in our life as a living organism, as a human. So I hope that you learn something in this video, and see you again in the next video, the next topic. So thank you so much for watching. Bye.